Hello everyone, I am Dr. Sneha, the faculty for OBGYN. In this video, we'll be discussing about implantation and placental development from Williams-based teaching. So, implantation is the first or the very initial starting point of any obstetric event that follows. So, when we look at this entire topic, we can discuss this under the following subheadings. We'll be looking at what happens just before implantation, how implantation occurs, the early embryo development, just the few steps of the first few days of early embryo development. We won't be looking at this too much into detail. Then we'll go on to the trophoblast development. This is what gives rise to the placenta. Then we have the amnion and the umbilical cord development, and we'll be ending with the functions of placenta. So let's look at a timeline of what happens in a woman's menstrual cycle. So let's say that the woman has a regular menstrual cycle. We expect around the 14th day that ovulation occurs. So this is when the egg or the oocyte is released from the ovary. And around this time, if she happens to have intercourse and there is fertilization, it will lead on to the formation of a zygote or the early embryo. Now, around 1 week after the fertilization event, around 22nd to 25th day to be precise, this zygote travels through the tube, enters the uterine cavity. Around this time is when you will expect the implantation to happen. Implantation is the process by which this embryo, the early embryo, sticks or attaches itself very firmly on to the uterine surface. Now, around the 28th day following this is when you will expect the next periods to come. But if at all the pregnancy has happened, she will miss her periods at this point. Now, following the implantation, the following next few days is going to be very important for the development of the placenta and the early embryo. In fact, the entire placental circulation or the earliest form of fetal circulation is established as early as 17 days from fertilization. So let's take the date of fertilization as day 0. So from the 17th day, so that would be around 31st day of your starting from your LMP. So around 31st or 32nd day, in this time the fetal circulation along with the placenta is formed. The primary form is formed. So we are going to look at a series of events which happens during this around 2 weeks time. Even though it is a very short period, a significant and very important events happen during this time. So let's look at it one by one. We'll start with the pre-implantation events. As I mentioned, on the 14th day, ovulation occurs, and once the ovulation occurs, the Graafian follicles, under the influence of the luteinizing hormone from the maternal pituitary, will undergo a process called as luteinization, where the Graafian follicle is converted into corpus luteum, a structure called as corpus luteum. This corpus luteum plays a very important role of producing the progesterone hormone. If at all fertilization happens, in the initial few days, the trophoblast cells or the initial placental cells produces beta HCG, which is going to stimulate this corpus luteum to produce an important hormone, progesterone, in the initial few weeks till the placenta takes over this function. So the corpus luteum is a very important structure to be formed if at all this is going to be an ongoing pregnancy. If there is no fertilization that occurs and if she is bound to have the next menstrual cycle, this corpus luteum will eventually become a fibrosed scar tissue called as corpus albicans. If there is no pregnancy happening. Now, what happens to the endometrium following this? So, around day 22 to 25, that is approximately a week or so after the ovulation happens, the endometrium readies itself for receiving the embryo. So, during this time, a lot of secretory changes happens in the endometrial layer. The glands become more coiled to increase their surface area and also becomes much bigger in size, and these glands starts producing more significant or copious amount of secretions as well, and you might notice something called as pinopods on the surface of this endometrium. So the endometrium is covered by epithelial lining which has small microvilli on them. During this period, 1 week after fertilization, a couple of these endometrial epithelial cell will fuse together to form small flower-like projections. Now, these act as a sticky tape to receive the endometrium, to stick for the embryo to get attached to the endometrium firmly. These pinopods are formed. So this is how a pinopod will look like under electron microscope, very pretty flowery appearance. Now looking at the vasculature, the spiral arteries also, just like the glands, will undergo a lot of coiling, and there will be increase in growth as well to make sure that the endometrium is richly supplied with the vasculature to provide adequate blood supply to the embryo that it is going to receive. Now this period where all of these important changes occur is the best time for the embryo to get attached itself to the endometrium. So that is why this is called as the window of implantation. Now if pregnancy happens, if the embryo is received by the endometrium, now it is no longer called as endometrium, but the lining of the uterus is now called as decidua. Now decidua is nothing but a modified endometrium that you see during pregnancy, and this decidua is given a different name depending on which part of the uterus or the embryo it covers. This decidua helps in the formation of human placenta, which is described as hemochoroidal placentation, which means it facilitates the interaction between the maternal blood, which is the hemo, and choroidal, which is the chorion or the chorionic villi, which is formed from the trophoblast. So the maternal blood, which is the maternal component, and trophoblast, which forms a placenta, which will be an interaction with the fetus. So this decidua helps in the formation of a hemochoroidal placentation. Now let's look at the parts of decidua. There are 3 decidua parts depending on which part or which layer of uterus it covers. The first is called decidua capsularis, which is the decidua that overlies the embryo that has implanted into the uterine wall. So this brown is the myometrium, the pink is going to be the endometrial lining. Now we are going to call it as decidua, and you can see the embryo that has implanted itself into the decidua, and this pink layer of decidua that covers the embryo is called as decidua capsularis. The next is decidua parietalis, which is the decidua which covers the rest of the uterus. So the entire layer which covers the other parts of the uterus is called as decidua parietalis. The third is decidua basalis, which is the decidual layer which is seen just below the implanted embryo. So that is, the green part is decidua basalis. Now the decidua capsularis and parietalis are together given a name called as decidua vera, and decidua parietalis and decidua basalis, only these two, is again divided into 3 layers from top to bottom, not Decidua capsularis, because Decidua capsularis gets compressed by the growing fetus and it just becomes a thin sheet of film, whereas the parietalis and basalis continues to grow. So there are 3 layers starting from top to bottom, just like how we have seen the endometrium having layers. There's going to be zona compacta on the top, zona spongiosa just below it, and zona basalis. The compacta and spongiosa are going to called as zona functionalis. Okay. Now, the decidua in pregnancy is very special because it is capable of producing the prolactin hormone, and this prolactin hormone that is secreted by the decidua has 3 important functions. First point is that it is proangiogenic, which means it helps in the formation of new blood vessels where the fetus gets attached to help with increased blood supply. Second, this prolactin can get cleaved by the protease enzyme, forming a substance called as vasoinhibin, and this vasoinhibin is now found out to be one of the important pathogenic agent for the woman to develop peripartum cardiomyopathy. Third function of this decidual prolactin is that it decreases the insulin activity in the mother. So it leads to insulin resistance. In our physiology video, we saw that insulin resistance is a very important physiological process that occurs during pregnancy, which helps in the fetus to get a sustained glucose supply. So the decidual prolactin helps in all of these functions. Now, as the fetus starts growing, this space between the growing fetus and the rest of the uterus gets smaller and smaller as the fetus grows. So it keeps expanding and at one point this decidua capsularis and decidua parietalis fuses with one another when the fetus grows to a certain size. So this fusion happens around 14 to 16 weeks, around the beginning of the second trimester. Let's go on to the next event. We are going to see how implantation occurs and how trophoblast is formed. Let's start with fertilization. So now during ovulation, we know that the ovary releases out a oocyte. Now this oocyte is called as secondary oocyte, the one that is released by the ovary during ovulation. It has completed primary meiotic division, hence it is called as secondary oocyte. When it has completed primary meiotic division, it has released one polar body, which you will be able to see. Now it is in the process of undergoing secondary meiotic division, but it has not completed yet. So it is undergoing secondary meiotic division. If the woman has intercourse during this ovulation period and if the sperm fuses with this secondary oocyte, fertilization occurs. Now during this fertilization, the secondary meiotic division of this secondary oocyte is completed, which will lead to extrusion of the second polar body. So you will be able to see two polar body. Now this fertilization, or the part where the sperm goes and inserts itself to the oocyte, is helped by a membrane or a layer around the oocyte called as zona pellucida. Zona pellucida has a couple of important functions. The first being it helps in acrosomal reaction of the sperm head. So this helps for the sperm head to lyse the oocyte covering to get inside the oocyte to fuse. Second is that this zygote that is going to get formed has a very high propensity to get attached itself to the walls of the tube before it reaches the uterus. So the zona pellucida acts like a film or a layer that prevents the stickiness of this early embryo, prevents sticking or attaching of this early embryo into the tubes. It helps to maintain the embryo to be traveling through the tubes till it reaches the endometrial cavity. Third is that these zona pellucida do not have any major histocompatibility antigens which are responsible for rejection of any foreign bodies. Now this embryo is formed by the fusion of maternal and paternal cells. The paternal cells are going to be completely new to the mother, which means that this embryonic cells are also partly foreign to the mother. You don't want the mother's body to reject this embryo before it attaches itself or implants itself. So the zona pellucida acts as an important factor to prevent rejection of the developing embryo. So these are the three important functions of zona pellucida. So once fertilization occurs, it leads to a two-cell, the beginning, the most primitive form of the developing baby, which is going to be the zygote. The zygote is going to have a female pronucleus formed from the oocyte and the male pronucleus which is formed from the spermatozoa. This zygote will keep dividing itself exponentially from 2-cell to 4-cell to 8-cell to 16-cell and so on, and keeps growing in number of cells, and at one point it will go to a 16-cell stage. This is called as morula, and it will look like a mulberry with lot of small projections around it. You can see that it is still covered by the zona pellucida. Now this whole process of being in the tube till the morula stage might take up to 3 days post fertilization. So from now on, we are not going to talk about the gestational age from LMP like how we usually say in obstetrics, but we are going to talk about the timeline is going to be explained post fertilization days. So 3 days post fertilization, the early embryonic development is going to happen in the tube, and then around the 4th day it enters the uterine cavity. And can you see a small change or a difference in structure of the morula? You can see that there is some amount of fluid that is accumulated inside the morula, which has divided or pushed the inner cells into one corner. So in one corner you can see a clump of cells, and this part is called embryoblast, and there is a thin layer of cell that has been pushed to around the structure. So this is called as the trophoblast. Now this embryonic structure, which is formed by the accumulation of fluid in the morula, is called as the blastocyst. The formation of blastocyst happens around 4 to 5 days post fertilization, and this is when it enters the uterine cavity. And now this blastocyst, once it enters the uterine cavity, as you can see, it loses the zona pellucida, which means that now it is ready to get itself implanted to the uterine cavity. Now let's look at the implantation process. We know that a blastocyst is formed and it is inside the uterine cavity. Now implantation happens 6 to 7 days post fertilization, and this is brought about by 3 major steps. The first is apposition, which means that this blastocyst comes in close contact with the endometrial lining. Now this is brought about by certain factors which is released by the endometrium, such as CSF, LIF and follistin, and also by a factor or a hormone that is produced from the embryo the trophoblast to be specific, which is the beta HCG. Now both of these substances that is secreted by the maternal endometrium and by the developing embryo attracts each other like magnet, which leads to the attraction or the makes the embryo to travel in close proximity to the endometrium, which is what we called as apposition. The next is adhesion, where when they come next to each other, they stick. The blastocyst sticks to the endometrium, which we call as adhesion. So this adhesion is brought about by other molecules that is again secreted by the trophoblast cells of the blastocyst, called as cellular adhesion molecules and integrins, which helps the blastocyst to stick to the endometrium. The third is invasion, where the blastocyst completely burrows itself inside the deciduum. So it is completely surrounded by the decidua. So that is invasion. So implantation happens through these three stages. So if there is a mismatch between this endometrial receptivity or the decidual receptivity and the day the embryo is transferred into the uterus, for example in IVF pregnancy, if the transfer is done a few days earlier or few days after, before the time that the decidua is ready to or the endometrium is ready to receive this, it can lead to repeated implantation failure in IVF. This could be sorted out by looking at the endometrial receptivity. So this is a test to check if the endometrium is ready to receive the blastocyst that you're going to put inside the women's uterus during IVF. Now let's look at just a few steps of how this embryo or how the blastocyst further develops into an embryo. Just a few steps, because this is important to understand how the trophoblast further develops into a placenta. So we saw that the blastocyst fluid inside, and there is a clump of cells at one pole. This part is called as the embryoblast part, and this part is called as the trophoblast. The cell that lines all around is called as the trophoblast. So two cells. Now this further develops to form the embryoblast further divides itself into two types of cells. The upper part of cells become taller, columnar-like, and this layer is called as the ectoderm. And the lower cells become flattened, and they are called as endoderm. The fluid is still there, and the yellow cells or the trophoblast. Now next, you can see that one more cavity has formed just above the ectoderm, and this cavity is lined by this green cell. So this green cell is called as amniocytes. They are to become the amniotic membranes in the future. Okay, this amniocyte is developed or it arises from the ectoderm cells of the fetus. So the ectoderm gives rise to amniocytes, which is going to form the roof of this new cavity, and this cavity is going to be the. Now another change that is happening is that you can see that the endodermal cells have actually lined the inner part of the fluid cavity, and this membrane is called as the Huesner's membrane. Now the origin of this Huesner's membrane is a little controversial. Some people say it is derived from the endoderm, but some people say it might be from the trophoblast. But this Huesner's membrane is going to line this part of the cavity which contain the fluid, and now this cavity is going to be the primary yolk sac. Okay, now let's go on to the next development. Here first let's orient ourselves. The outermost layer is going to be the trophoblast cells. We have the tall columnar cells on the top, which is the ectoderm. Then we have the smaller cells in the bottom, which is called endoderm. We have the amniocytes lining the ceiling of the cavity on the top. This is going to be the amniotic cavity. Then we have another layer around the primary yolk sac. Now what is new here is this pink structure that divides the inner embryal cells from the trophoblast. Now this pink layer that you see is called as the extra-embryonic mesoderm. Now this extra-embryonic mesoderm, or a group of cells that are produced inwards from the trophoblast, and it separates the trophoblast from the inner embryonic mass. It's called extra-embryonic because it is outside the actual embryonic structure and it does not give rise to any of the fetal parts. After this, small spaces, empty spaces starts developing inside this extra-embryonic mesoderm, and these spaces coalesce or join with each other to form a cavity inside the extra-embryonic mesoderm, which is called as the extra-embryonic coelom. Alright, so now you can see that the entire extra-embryonic mesoderm has been split into two layers by this cavity. The inner layer is now called as the parietal layer of extra-embryonic mesoderm. The inner layer is now called as the visceral layer of extra-embryonic mesoderm, and the outer layer is called as the parietal layer of extra-embryonic mesoderm. Now you can see that the entire embryonic structure, the inner embryonic structure, is suspended from the trophoblast by just a small tissue of extra-embryonic mesoderm. So it is hanging in this cavity of extra-embryonic coelom, and this part of extra-embryonic mesoderm which connects the embryo to the trophoblast is called as the connecting stalk. This connecting stalk will later go on to form the umbilical cord. Now once all these changes happens, the primary yolk sac becomes much smaller, and now it is termed as the secondary yolk sac. So these are some of the very early embryonic changes that we should know before we dive into the topic of trophoblast development. Now let's look at the trophoblast development, the actual development of the placenta. These are the original cells that are going to give rise to the placenta. The placenta is going to function like the fetus's lungs, the liver and the kidney. It's a very wonderful organ with a lot of important function. Now this trophoblast development occurs around 8 days post fertilization, immediately after the implantation happens. The trophoblastic cells are divided into two types. First is the cytotrophoblast, which is the layer of the outermost layer of the trophoblast. These have proper cellular structures with a cell membrane. The second is called as the syncytiotrophoblast, which means that they don't have any cell membrane, but they are bound by syncytium, so they can move around or they can invade into the decidua very easily, and they just have nucleus scattered all over them. They don't have a typical membrane bounding them. So these are called as the syncytiotrophoblast. So this is how the trophoblast types are divided. Now this syncytiotrophoblast and cytotrophoblast is again divided into two types based on where these trophoblasts invade. The first is called as the villus trophoblast. So these are the trophoblast that is going to develop into the chorionic villi, which are the major structures of the placenta, which helps in the oxygen and nutrients oxygen and nutrition exchange between the mother and the fetus. The second type is the extravillus trophoblast. As the name implies, these are the trophoblast that helps in structures or helps other than the chorionic villi formation. So these are trophoblasts that invades the decidua and the myometrial spiral arteries, the maternal blood vessels. So there are two types, interstitial, which burrows into the decidua, and these give rise to placental giant cells. The second is the endovascular type of extravillus trophoblast, which invades into the spiral arterioles, which we know are the end part of the uterine artery, the one that supplies the uterine musculature and the decidua in this part. So the trophoblast that invades the spiral arteries are going to be called as the endovascular trophoblast.